In this video lesson, we will study about nucleic acid. Nucleic acid history, its structure, its types, and its functions. In 1868, Swiss physician Friedrich Miescher extracted a novel substance from the nuclei of white blood cells, leading to the discovery of nucleic acids. This substance was a unique form of biological molecule because it was neither a protein, lipids, nor a carbohydrate, because he had isolated it from cell nuclei. Miescher gave his finding the name nuclein. He made what is today referred to as deoxyribonucleic acid. Nucleic acids are large macromolecules, which is necessary for all organisms and viruses to function. The same elements found in proteins, including carbon, hydrogen, oxygen, nitrogen, and phosphorus, are also found in nucleic acids. Nucleic acid are polymers that consist of nucleotide residues. Nucleic acid are located in nuclei of cell, hereditary determinants of living organisms, hereditary factors that affect how living things behave. Nucleic acid can be defined as a complex organic substance present in living cells, especially DNA or RNA, whose molecules consist of many nucleotides linked in a long chain. There are two primary forms of nucleic acids, that is, DNA and RNA. DNA can be defined as self-replicating material that is present in nearly all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. DNA is made of two linked strands that wind around each other to resemble a twisted ladder, a shape known as a double helix. Each strand has a backbone made of alternating sugar deoxyribose and phosphate groups. Nitrogenous bases are linked to each sugar. Ribonucleic acid, also known as RNA, is a type of molecule that exists in every living cell. The primary role of this is to convey DNA messages that are responsible for controlling protein synthesis. RNA is composed of nucleotides. The components of nucleotides include sugar, that is pentose, bases, and phosphate groups. The nitrogenous bases are purine bases, that is adenine, guanine, and pyrimidines, that is uracil and cytosine. The structure of nucleic acids, like DNA and RNA, is referred to as nucleic acid structure. Chemically, DNA and RNA are quite similar. The four levels of nucleic acid structure are primary, secondary, tertiary, and quaternary. The primary structure of a nucleic acid refers to the linear sequence of nucleotides along the polymer chain. For DNA, this sequence is composed of the four nitrogenous bases adenine, cytosine, guanine, and thymine. For RNA, the sequence includes adenine, cytosine, guanine, and uracil. The order of these bases and the sequence carries the genetic information. Unlike proteins, nucleic acids lack a secondary shape. However, they can exhibit specific interaction patterns which give rise to a secondary structure. The two DNA strands wrap around each other in a spiral shape. The building blocks of DNA, called nitrogenous bases, join together with hydrogen bonds in a specific way. Adenine always pairs with thymine, and guanine always pairs with cytosine. This matching creates the steps in the DNA ladder, while the sugar phosphate parts create the sides. The tertiary structure of nucleic acids refers to the three-dimensional folding and interactions of a single nucleic acid strand. This folding is influenced by various interactions, including hydrogen bonds, base stacking interactions, and interactions between different parts of the same strand. Tertiary structure is crucial for the functionality of RNA molecules, especially those involved in catalytic and regulatory functions like ribozymes and regulatory RNA. Quaternary structure refers to 3D interactions between multiple subunits in the case of nucleic acids. Quaternary structure refers to interactions between multiple nucleic acid molecules, or between nucleic acids and proteins. Nucleic acid quaternary structure is important for understanding DNA, RNA, and gene expression because quaternary structure can impact function, for example, when DNA is packed into heterochromatin. Therefore, exhibiting a type of quaternary structure, gene transcription will be inhibited. Each type of nucleic acid carries out a different function in the cells of all living things. DNA stores the genetic information. The information is transmitted from parents to children through the process of DNA replication. When DNA is replicated, it makes a duplicate copy of its DNA. Then the cell divides, resulting in the correct distribution of one DNA copy to each resulting cell. The primary function of RNA is to create proteins. By a translation, RNA carries genetic information that is translated by ribosomes into various proteins necessary for cellular processes. Not all nucleic acids are involved in processing the information stored in cells. The nucleic acid adenosine triphosphate ATP, made up of an adenine nitrogenous base, a 5-carbon ribose sugar, and 3 phosphate groups, is involved in generating energy for cellular processes. Thank you for watching, and don't forget to subscribe, like, and share.